a joint seminar between the Waterways Centre for Freshwater Management and the School of Biological Sciences. It's my very great pleasure to introduce Dr. Catherine Febria, who many of you will know. Catherine's been here for I think 4 years now. Just ticked over 4 years. It arrived in 2012. We've been really lucky to have her. She's been a great mentor and leader in the freshwater scene here, and Catherine's been leading as a sort of director of the CAREX program, which I'm not going to say anything more about because Catherine's going to introduce that. But you've also been busy with quite a few other things, including an IPBES fellow, which has been an important role. I'm not sure you're going to talk about that at all. Catherine came here from the University of Maryland, where she was working on other restoration projects and did her PhD, I have to make sure I get this right, in 2010 at the University of Toronto in Canada. And you're actually returning to Canada to take up Canadian Research Chair, which is a really prestigious position. Catherine's going to be leading a team at the University of Windsor's Great Lakes Institute for Environmental Research, and that's about the restoration of headwater streams, which we're really delighted about. It's a fantastic opportunity, and we're very lucky to have had you for 4 years. We're going to be sad to see you go, but we know that you're going to keep doing great stuff, and we're really looking forward to hearing about what you've been up to over some of that time.
Great.
Thank you.
Kia ora tātou, iti mana whenua tēnā koutou. Ko Catherine aho, and I wanted to thank Angus for the introduction and Jenny and the Waterway Centre for just this generous support for the seminar. So I've thought about what I wanted to talk about today, and a lot of it has to do with this idea and this research I've been doing on small streams. And really the challenge of trying to address a lot of these complicated environmental problems that we have today, but by tackling them, big problems in small ways. So I wanted to first just the type of researcher and the type of scientist that I am has really been about making connections and thinking about the big picture and trying to address them in small chunks. And some of you might recognize this area. Does anyone from here or affiliate to this area? And this is I'll just wait a minute for folks to come in. Kia ora. And so a lot of my journey in the restoration space and the freshwater space really started in this corner of the world. And so this is the Great Lakes of North America. I spent a lot of time in this little pocket over here. I wasn't born there. I immigrated from the Philippines to Toronto, and I spent a lot of time on the Great Lakes. And a lot of that time was focused on how we couldn't swim there, and the lakes were unhealthy, and there were these things like acid rain and other issues around the world that was up to my generation to solve. And so that's a really big thing, as you're a scientist coming through the system, thinking about what can you do to make a difference. And so I really kind of thought about what I wanted to do with this toolbox and this skill set that I was really attracted to in science. And so I looked to the elders in the scientific community like E.O. Wilson, and around in the 90s there were essays, beautiful essays that he would, and reflections and things that really resonated with me were comments like, this next century, I believe, will be that of restoration and ecology. And so it was really interesting because I realized maybe this is the kind of work that I want to do. At the same time, E.O. Wilson, as many of you know, has done a lot of really interesting work with ants and is famous for a lot of his writing on nature and the effect of people have on this planet. He also said things around the same time of, if I could do it all over again, I'd be a microbial ecologist. And so I thought, oh, that's really interesting. What does that mean? And what kind of, what do I want to be in this landscape? What kind of scientist do I want to be? And so a lot of my elders in the community at the time said, if you want to understand how Canada's freshwaters are changing, go to the north. And so I spent a lot of time in the Canadian Arctic, and this is a photo of the Mackenzie River floodplain where 50,000 lakes are connected in multiple different ways. It's a beautiful natural outdoor experiment to think about the relationship of streams and rivers and climate and communities. And so it's in this time where I really thought about the microbes. I thought about carbon, and I thought about how this changes in response to things like climate change. At the same time, I worked alongside different communities, Dene communities, Butchin communities, scientific researchers who were trying to grapple with how Canada's ecosystems were changing. There were really interesting and profound moments where some traditional elders and ethnobotanists would collect plants and talk about what those words were and how they harvested them. But then they also had observations about how things like the North American robin were coming up to these regions, and they didn't even have a word for that in their language. And so within a generation, the world as they knew it was changing. And so that's those are sort of threads that I've carried with me in putting together and pursuing research that I think is interesting, at least to me, and helpful in solving the problem of big environmental problems. So there are 3 things that I hope you take away from today. I know this is a big mix of people from social scientists to practitioners and students and ecologists. And I hope to show you that over the last 5 years in particular, the research that I've done have addressed 3 things. One is that small streams are powerhouses. I love them. They're transitional, they're dynamic, they're really hard to sample, but it's in these systems where we realize that they're driven by the things you can't see. And the second thing is that working in these waterways and these small places is actually where the places where people live and where they work. And so what that has done has made freshwater scientists kind of move across disciplines and learn and move in this place called translation ecology. And one of the biggest issues that I see in this field is trying to address risk and uncertainty in the way that we manage them. And thirdly, because I'm interested in connectivity across scales and across disciplines, we just need to move from think about our local places, but not lose sight of that big picture and know that they are all related and interacting. And so I'll give you some examples at the end, if there's time, about how we've been able to do that here in biological sciences and at UC. So I first wanted to start at the beginning. Again, as a young ecologist, I was really, impressed. I was very impressionable. I would read these things by scientific elders, and I first started with reading some of the original works, and one of them was Arthur Tansley's essay where he defined ecosystem. What are we even talking about? And if you read this essay from 1935, Tansley talks about how ecosystems vary in sizes from the molecular atom up to the entire universe. That was really profound for me because I thought we were talking about a lake or a river, but actually in its very first definition, he acknowledged that we need to isolate them mentally, but they interact and they overlap and they interact with one another. And then as a discipline in ecology, we needed to proceed in isolating them sort of artificially, but there was a recognition even in its first definition that these ecosystems are very tightly interacting. And I think I realized then is that the interactions and the connectivity was the space and the gaps that were biggest in freshwater, and sort of the gap that I wanted to try and fill. So again, I go back to the headwaters. I spent a lot of time in these rivers. We probably all have spent time in some type of waterway. And a lot of great thinkers in this system, in this field, have identified why headwaters are so important. We know that Judy Meyer's essay or paper from 2007 just reminded us that headwaters are essential for organisms that live in lakes and large rivers and oceans. We have fish that come up and spend critical components of their life cycle in these places. And they take refuge in these headwaters. There's because of all sorts of reasons. It's the thermal conditions, the redox conditions, community interactions, and so forth are different in the headwaters than they are downstream. And equally, invertebrates carry out essential portions of their life cycle here. They interact with the landscape around them, and the landscape around them generates nutrients and all sorts of organic matter that are taken up in the food web in multiple different ways. And if we dig down even further at the interface of in the sediments of these headwater streams, there's an even richer community still in between those tiny interstitial spaces where you have microorganisms interacting with different gradients in oxygen. And so this, at whatever scale you're looking at, there are these complex interactions that we still have very little understanding about. So I kind of scaled back up to the watersheds, to the Great Lakes level, and I realized, for all of these large lakes, there are all of these watersheds that we have no knowledge about. We know what's happening in the larger rivers, but those real connections between the land and the water are these sort of, seem like a really glaring gap. And so I spent a lot of time just kind of thinking about and identifying who was doing work in this area. And in particular, I went to see these people, Dudley Williams in particular and Noel Hines. They spent decades of their life defining and establishing just how important streams were and their connection to the landscape, how important that was. Noel Hines has one of these most highly cited paper talking about how a stream is intricately defined by the catchment around it. It's influenced by the surrounding catchment. And Dudley Williams, who would later become my PhD supervisor, did a body of work very systematically and carefully on the invertebrate communities in this place called the hyporheic zone. You know, this is where the party is happening if you're in a stream ecosystem. You live in the benthos, the highest rates of biomass are here, the highest rates of organic matter are here, you have these rich gradients in oxygen and other sorts of nutrients, and that was a great picture, or a long body of knowledge here in the Great Lakes were from this lab and these people. So I hung out with them for my PhD, and I realized that I could spend a lot of time addressing some fundamental gaps on how an environment like the headwater ecosystem could change the structure and function of headwaters. And maybe one day that would link to how we could effectively manage them. So I went back to the same sites where Noel Hines and Dudley Williams did a lot of their work on the hyporheic zone, and I went further upstream to the headwaters, to the intermittent sections of where a lot of that work was conducted. I Swiss cheesed all these intermittent streams, and I tried to measure all sorts of parameters, and I wanted to build on that body of knowledge that Dudley and Noel carefully created. I looked at the bacterial communities because that seemed to be the biggest gap in terms of linking the landscape and nutrients to the invertebrate communities that we had a really good understanding for. And in particular, I worked with bacterial communities and extracted the 16S gene. I used a method at the time about terminal restriction fragment TRFLP, which looked at the relative abundance of bacterial OTUs and their abundance based on this fluorescence. Now, I sampled these communities over different transitional gradients. Intermittent streams, permanent streams, drying and wetting events. And if you can imagine a stream going from your left to right, these headwater streams would have these really strong gradients. One week to the next, we might see it completely dry, and then within a couple of days, completely flooded. So I sampled these things and found that unlike the invertebrate communities, which we knew were really dynamic, the microbial communities were even more dynamic still. So within a few days, we saw community composition shifting on the scale of days. I also explored the carbon itself, and we have these beautiful technologies that can draw little rainbow topographic maps about the molecular structure of carbon. And so by looking at the fluorometric properties of it over time, we could then generate a better idea of what the carbon community looks like. And in that way, I spent, I really enjoyed this time. It was a really productive time of thinking about intermittent streams, structure, function, and generating new knowledge on what headwater streams look like, at least from the bottom up. But at the same time, I did a certificate in environmental studies. So in addition to doing a lot of field work, I spent a lot of time with climate change scientists, with philosophers, with lawyers and engineers, and we were forced to talk about environmental issues. Our course assessments were all about talking about issues that we talk about all the time. What do we do with our rubbish? What do we do about chlorine in the water? All of these things, and we really need to dissect them from all particular angles and think about what is the role of science in that landscape. And so it was really clear then to me that I don't know how the microbial data are going to answer that question, or I just really was trying to find the connect. But one researcher really stood out. As I continued to kind of read about stream restoration, the same authors kept popping up, and in particular this one came up. Every paper that I would read was really clear to me. It just seemed like a light bulb every time I'd read these papers. There's this thing called the Field of Dreams hypothesis, named after the Kevin Costner's movie, for those of you who are old enough to remember. This idea that if we create habitats, species will come. If you build it, they will come. Invertebrates and fish, just add the habitat and they will come. And time again, this hypothesis failed. And it then, it sort of prompted a reflection in the restoration community, especially in streams and rivers, to say, say, what are we doing? We have no standards for doing this so that we're achieving ecologically relevant outcomes. And when we synthesized this at a larger scale, we realized that billions of dollars are being invested in reshaping rivers and reshaping streams with actually very little evidence to show that communities and functions and these ecological benefits that we're hoping will bounce back are ever achieved. And so this is like a multi-billion-dollar industry. We also learned from the same person that, if you completely blow off the top of a mountain and then recreate all of the stream somewhere else, that's actually a bad idea. And so what did I do as a PhD student, really naive in Canada? I emailed her. I was like, hello, Dr. Palmer, I like your papers. I think this is really cool stuff, and I'm going to be finishing my PhD, and I'm just, wondering if you have anything, if you have any jobs or opportunities and that sort of thing. Anyway, unbeknownst to me, in the south of the border, people were really up in arms around the protection of temporary streams. The Clean Water Act and various legal things that were happening were suggesting that temporary streams were one of those types of ecosystems that were continually being buried. Maybe up to 77% of small streams in urban areas were being buried with no legal ramifications. One of the senators, or one of the judges in the hearing, said, well, if you can establish a critical nexus between a temporary water body and a permanent one, then we might be able to review this decision. To make a long story short, the science community kept arguing we need to value and protect these temporary waterways, but in the lack of any sort of real evidence, to make a long story short, it was vigorously debated. In the end, the Clean Water Act eventually identified intermittent streams in their legislation, but that's kind of changed since the last administration. So we can talk about that over tea or beer, but anyway, it turns out that this was an area of science, an area of policy that really needed some good science. So I worked, at the same time I should acknowledge that Margaret then ended up becoming the director of a national socio-environmental synthesis center. This was a great place for me as a freshwater ecologist to talk to economists and social scientists and other scientists about these kinds of things happening on landscapes. And at the same time, while she was getting that up and running, I was able to mentor PhD students, and one in particular, Jake Hosen and I, decided that we were going to use his PhD to sample the wazoo out of this particular catchment. We wanted to see if we could measure all sorts of microbial and molecular kinds of indicators of ecosystem processes. We found a catchment that was almost 100% forested except for a little bit of urbanization up here, and we thought, well, if we sample the intermittent and the permanent waterways, if we sample the water and the sediment, just with the strength of number of samples and time and seasons, we should be able to try and understand the significance of headwaters in these catchments, the impact of urbanization over time, and that's what we went about doing. So some of the early work, so the two students I worked with, Jake and Owen and I, we sampled this catchment, and we thought about how does urbanization change the structure of carbon and microbial activity over time. And what we were able to show, and so this is the graphical abstract from that, so if you were to take a watershed, add some sort of urbanization on the landscape, how would that change the molecular structure of carbon, and how would that change the microbial bioavailability of carbon and nitrogen and their activity rates? So you can do things like, so with the power of 500 samples, we were able to take all of the molecular signatures of carbon and create a model. And you could take all of those samples and dilute or predict that all of the carbon was driven by 5 main types. And so they are C1, C2, C3, C4, and C5. You could then go visit the published literature to places that have isolated them and identify that if you have a carbon signature of this excitation and emission, it was most likely linked to, in C1, forest leaves, terrestrial material. That would make sense. You would also maybe link it to things like tryptophan, which might be associated with things like biofilms. But also in this watershed where we had such little suburban development, like 8% of the watershed was suburbanized, we identified 2 strong, significant components linked to urban or anthropogenic land use. And so that was really interesting for us because then we were then able to relate some of these changes to microbial activity rates and the community structure. So If you think of, so we looked at the molecular community structure, and in this case, we're looking at the 16S community, and we looked at the temporary waterways and the permanent waterways. And I think from all accounts, at this course level, if you just look at the color boxes, they look pretty similar. The same types of taxa were there. But then when we actually looked at how those communities related to each other across ecosystem types, we see a completely different picture. So what we're looking at are the same taxa, but each of them are now connected in this network analysis. Two things will pop out. The permanent streams are in blue, and the temporary streams are in red. And there's very little association in the microbial communities over ecosystem type. What was also really interesting is when you actually look at the taxa that are there, we noticed that there were certain organisms, or certain groups that were more closely associated with the permanent streams, and in particular carbon fixers and the nitrospiralis group in the intermittent stream. You know, there's some suggestion that you can use these kind of tools to identify keystone taxa in ecosystems. And so we were left kind of with this study thinking about, could we use these tools and this tool set to figure out what are those keystone functions of permanent streams, and it's most likely driven by these microbial communities. So, like I said, Jake and I have gone and done, we have lots of great ideas for how you can move this further, but in addition to looking at the molecular and the headwater sort of ecosystems from the ground up, I was also looking at the world around us, which was these environmental issues around how you manage these waterways, recognizing that despite, you know, the watershed that we were looking at, which was highly forested, the rest of the Chesapeake Bay was featuring or being subject to a completely different situation. And so that kind of leads me to the second piece of this type of methodologies and research that we did that wasn't about generating new science, but trying to synthesize the existing science and knowledge that was available for the region. So I want to zoom back up and think about that watershed. The watershed that Jake and I worked on was just right over here. A small little watershed, but the Chesapeake Bay larger watershed is huge. It's like 64,000 miles across or something like that. It's 5 states and the District of Columbia. I mean, the issues that we were facing with, you know, that it was really, you know, we were grappling with how do you make what we did in this little Parker's Creek watershed useful to a decision maker. Well, thankfully, EPA, they had questions for us, and they came and said, well, what are the roles of watersheds in restoring Chesapeake Bay values and issues? We're worried about fisheries, and you know, and we're not worried necessarily in the forested ones. I think, you know, they're forested and those are protected. What about the ones where people are living, these urban catchments, and how well are they working? So that's a completely different question, you know, what we thought scientifically is really cool versus what the decision maker actually wanted. And so we spent a lot of time talking to social scientists and economists about this thing called ecosystem services. You know, it's a framework that all of us have heard about for over a decade now. And so I won't go into it in too much detail, but I'm sure you know about it, but you can think about that this is the way that humans value nature. It's about what we get from the headwaters: clean water, you know, cultural, recreational, sporting, you know, pleasure, and from these places. It might provide flood control and that kind of thing. And for many communities, this is where we gather our food. As an ecologist, I work in this space. You know, I'm looking at all those interactions over time. I think about, you know, there's biodiversity as a component of it, there's water filtration as a component of it, flooding and that sort of thing. And then there's this other component that's really tricky called restoration. We know it doesn't work that well, but we're spending a lot of money doing it anyway. How are these things related? And I was really happily in this box. And so I really needed to kind of scale and think differently about how to make those connections work. And a lot of that was through the power of synthesis. We first started by asking a second question. Before even answering that question to the EPA, we first needed to take stock of what we knew. Thankfully, Fish and Wildlife, who spends millions of dollars giving out money to restoration projects, was in the mix there. And they asked us, well, what kind of restoration is even happening? You know, what are the best practices for rivers and coastal wetlands? And, you know, what should we measure? Because, you know, if we're going to be giving out millions of dollars every year, we probably should all do the same thing, right? And they kind of needed that kind of validation about what we do, what we're restoring, and how do we measure if it's actually working to justify why they're spending millions of dollars on these sorts of projects in the Bay Area. So we did that. We reviewed 500 studies across the Chesapeake Bay. We looked at, for streams and rivers, what kinds of things people were measuring, what kind of services they identified, and from all of those research, we identified that from this laundry list of metrics, there was no consensus for streams or rivers on what sorts of things we should measure. Someone might be measuring temperature, and some might be measuring photosynthetic active radiation. And some people didn't even measure goals. And monitoring very rarely extended more than 1 or 2 years beyond a restoration-funded program. So that was a huge concern. And this wasn't just an issue in the Chesapeake Bay. This is an issue globally. There's really no consensus around how we go about restoring waterways. So knowing that, we returned back to the question that EPA posed us. What do we do about these urbanizing sites? People are living here. This is where, you know, we think we need to know how well our options are working. And we visited a place that the US Geologic Survey and EPA, the Environmental Protection Agency, had created and monitored for over a decade. And so if you look at this landscape, it looks like any suburban area that you might see anywhere in the world. And what we later realized is that it was embedded with all sorts of restoration structures in the design. And USGS and EPA had been taking a lot of water quality measurements over time. So we were able to harness that data and ask some questions about how well does restoration or these management structures work. We talked to the people that were actually modeling these things, so the hydrologic engineers, and we asked them kind of the same question. You know, if you looked at the literature, there was a multitude of types of models, SWOT, HEC-RAS, like so many acronyms, like you know, just all sorts of things that people were using to model how much, you know, nitrogen was coming off a landscape and ending up in the Chesapeake Bay. It was that simple. And we were able to identify that despite whatever model they used, it came down to these key things. What's the soil? How much rain is coming in? What is the slope of the catchment? How big is the watershed? How long has it been raining? What was the time since that last event? And what is the age and size and type of this management structure? So essentially they were asking very much the same question. In this environment, what structures are we placing and how well is it generating that function? The same paradigm of how are we influencing the nature of the ecosystem and the functions that we desire. So we were also told that there was plenty of data. You know, so we chose this case study because we knew there were a lot of data available. 700 based on a general literature search for very simple things about the most common stormwater management structures, nitrogen in particular, and we were looking for papers that had ecological relevant data, those things that the modelers told us were important. We also were told that there was a beautiful thing called this International Stormwater Database, 10,000 entries. We should be able to answer this question for EPA, no problem. We were only able to use 30 studies. And this was because they were the only papers that allowed us to calculate nitrogen coming in and out, because they provided discharge data, the size of the watershed, concentrations in and out, so we could go ahead and calculate them. And that was actually a real eye-opener for our decision makers. Here's one of those results. What you're looking at the bottom are the different stormwater structures that are normally used in an urban landscape. So dry ponds, wetlands, and swales for the 3 key constituents of nitrogen that people were worried about. I'll focus on nitrate because I think it's the one that's really interesting. And what we're looking at here is that black line, which tells us how much or how well does that structure remove nitrogen. So if it's above 100%, what you want is like 100% removal. Wouldn't that be great as you put, you know, you flush all of your nutrients down and whatever structure takes all of it out. But if we look at nitrate, it was about 30% effective at removing nitrate from downstream export. And so that's what managers are using in a lot of their planning. But when we actually include uncertainty and variability in that estimate, it looks like nitrate actually is plus or minus 100%, which is highly variable and usually overlooked or not even included in a lot of the management. So, you know, this was unsatisfying, obviously, and we wanted to see if we could do better to be able to answer the question that EPA posed, like how do we use this information to manage risk in the face of climate change? And we asked, can we use experts? Can we give more quantitative predictions? And so can we include uncertainty in the decision making? And one way that we were able to do this was to talk to mathematicians, and in particular Roger Cook. He's developed the Cook method. It's a method of including uncertainty in estimates of things when it comes to nuclear explosions and like things that are really high risk and really like big news, like the likelihood of ice sheets melting in Greenland and that kind of thing. So this seemed really low stakes kind of application for Roger, but he took it on, and he's sort of done the math to try and create an option for using experts to generate data. And so we used his method. We basically identified 10 stormwater experts in the region, people that really measured the ins and outs of nutrient loads across the Chesapeake Bay, with our partners at USGS and EPA who've been monitoring this particular watershed for years. We had some real data that we could ground those conversations around. We tried to incorporate the different geologic variability that we knew were important in the region. And we generated a 60-page or 60-question questionnaire for our experts. They were obviously compensated. They were up for the challenge. They thought it was kind of fun. But embedded in those questions were calibration questions, and that was key to Roger's method. So we gave data like this. So if you're a stormwater engineer, you love this stuff, apparently. So you love knowing that for any rain event, that you know how long it rained for, 14 hours, you know what the discharge was in the river, and how much precipitation. And if you know that, you can calculate nitrate loads, ins and outs. So we gave them 60 of these questions. Fun times if you are a stormwater expert. Rather than asking them, so the question was really specific. It was the same question 60 times over. I would say that the incoming load of nitrogen coming into this BMP there's a 50 chance of it percent being this kilograms, and sort of they define the distribution of that rain event for us. And they did this 60 times. We asked them what was coming in, what was coming out across a range of different BMP types, that sort of thing. We even peppered in some really extreme rain events to be able to get at that curly climate change question. What was really cool is that most of the experts, if for the calibration questions, if they knew what they were doing and they had their maths right, they nailed those calibration questions pretty well. We had one expert that was just off the mark all the time. And in a way it was kind of eye-opening, because he was one of these experts that's like, look, my business is on this type of BMP, and I'm gonna always give answers that will show that it works great. And that was, he kind of led with that. And so unsurprisingly, his estimates were always off the roof. So with Roger's method, we were able to kind of weight the experts based on their performance on the calibration questions, or give them equal weighting. Regardless, we had some data that we could then work with. To make a long story short, so I co-developed this with a biostatistician, Ben, and Ben modeled these with some climate change data that we were able to obtain from people like Catherine Hayhoe and Ann Stoner, who take downscaled estimates of climate change precipitation records. And we were able to tell EPA that the performance of the structures in the watersheds that they were worried about ranged widely, between 0 and 40%. Precipitation was the biggest thing driving the performance, and that the nitrogen loads, when we used these downscaled climate change precipitation data, generated higher, more variable loads under climate change. That is just not useful, but at least we were able to be quantitative about it and tell them that that the current things they have in place are inadequate for the future of climate change in the region, and that we really emphasize thinking about these conditions as events as distributions rather than the mean. So include uncertainty in your decision making, plan for risk, and and just make more informed decisions moving forward. So as you can imagine, that was like a mind, explosion for me working in this space. And, I really started in the freshwater space going into restoration. And as I realized, and as Succinct further developed, and I spent more time with the dark side talking to other people about, the same problem but from different lenses, this really kind of pushed me into this growing field called translation ecology. So I'm sure some of you have heard of it, but if you haven't, it is sort of this, there was a special issue by ESA about a year ago, and it was all dedicated to this growing field. what I think is really important about it and what I think resonated with me was that the realization that this is a whole kind of new and evolving field in ecology where we're spending a lot of time as a practice extending our research beyond theory or some opportunistic applications. We really are trying to build trust and trying to set ourselves up for doing research on a much longer term and really engaging science with their communities. At the same time, during that time, we've been reminded that what we need to do as a community is do more youth-inspired research, and that being driven by the needs of communities are really important here, and that things that my colleagues at CAREX were able to do was really focused on actionable science. How do you bring experts that to around a particular challenge and a particular issue, as opposed to collaborating in the same way, trying to get different results. So that's what brought me here. So, while some of that work from Maryland was kind of churning through, I was getting to know this region. And so let's zoom up. I'm sure everyone's probably looking, trying to find where they live on these maps and thinking about, where they go fishing and that kind of thing. But so here we are looking at the upper Canterbury region. And so I was really grappling with how can you kind of take all of this information about small streams and sort of things from the bottom up and thinking about what are the issues here in Canterbury. And how can we try and address the issues that people are really worried about? So a lot of it is driven by the fact that the landscape is shifted considerably in Canterbury, as you know, and the rest of New Zealand. We're thinking about in the Canterbury Plains how this was historically a swampland. It looks so different from anything that, from anything that it used to be. It's completely green. People tell me that this place actually was quite brown, and a lot of it in recent decades there's been a lot of concern about an intensification towards dairy farming and whether or not that is responsible for freshwater health in the places that we recreate, places that are important to us and identities as New Zealanders. And so at the same time, much like in other places in the world, the country is going through things like talking about what is acceptable for freshwater health, and so there was a national policy statement on this just a few years ago and that sort of thing. Okay, so I happened to join a group that had been working on this for a while. So Angus and John and their team had been thinking about surveying things like riparian management and all these restoration actions that people had underway. And it was clear from some of those early studies that small streams are really important to focus on. They're also not protected, they're in poor condition, but there was potential there. It also, for me, was a place that I thought was very actionable. An individual farmer, an individual homeowner could make decisions about how they manage their waterway if they knew that it was valuable and that it would make a difference. So if you remember that, so we took a similar approach to that picture I showed you before where we talked about values, and we knew we had a toolbox, and we kind of tried to design restoration trials to untangle and decouple and explore all of those kinds of interactions that happen in an ecosystem. And that's what we ended up calling CAREX, the Canterbury Waterway Rehabilitation Experiment. It was a mammoth effort that's been a lot of fun because it meant that we got to work with 23 or more farming families. We identified or created an experimental design that involved 9 different 1-kilometer waterways, and we trialed or thought up 14 different tools that had some realistic opportunity to be taken up by either a landowner, a farmer, regional council, industry. And so we initially engaged 40 stakeholders or decision makers in the different councils, and that number has really multiplied, and we've stopped counting in a lot of ways. And we all work together under this one umbrella called CAREX. The issues are complex, much like most freshwater ecosystems, and by talking to our communities, we realized that macrophytes was the main farming concern. There was a lot of flooding issues around that, and because of that, there's some relationship with sediment and nutrients, and ultimately this was undermining the freshwater ecosystem, the communities that were in these waterways. And so, what I'll say is that there was a lot of thinking about the scale at which these operate. And so we designed trials and trialed things at small scales and at larger scales to try and demonstrate potential solutions and generate data around what those impacts were. And one of them was around sediment. One of the biggest and easiest things we can do in the waterway is to prevent sediment from getting in, and that's called rebattering. And so it's been really powerful just doing it and showing communities that it's not that scary and that actually you're preventing further water quality be harmed by pulling these stream banks back and planting on them. We trialed things like sediment traps, and so if you dig a hole, it's very simple, and we worked with our district council's river engineers to do this and monitored the impacts over time. We were starting to learn that it's working really well. There's some practical implications to it. You need to manage it, but there's some additional unexpected benefits, such as creating habitat for fish and potential E. coli reductions that are apparent from this simple tool. So we found things and are learning things around how you can use tools to address the sediment issue. And that for the monitoring is helping us address that we need to also look at other potential trade-offs in the ecosystem. So something as simple as a sediment tool, we want to also look at phosphorus and sediment and trade-offs with E. coli and biodiversity. More complicated, maybe, is the weeds issue. And so Katie Collins, the PhD student who's recently finished, spent her PhD trying to find alternatives to the status quo. And so many of you will have seen her presentations over time, but in a nutshell, the current picture on the left is the status quo. This is how waterways are being managed for weeds across the region, and we surely need a better alternative to that for a number of different reasons. She did small-scale trials and large-scale trials and tried to identify alternatives to this. And in a nutshell, she found that things like riparian plants, CAREX secta in particular, if you choose plants based on their traits, they can outcompete these nuisance weeds that cause drainage problems. And that when you use them in combination, such as things like weed mat and spraying, you can boost that CAREX secta to grow and achieve full shading. And that might be a more sustainable option to the current regime. And of course, what it did, and when I sort of think about her research, it's really made us rethink riparian zones in general. It's a tool that most communities in New Zealand use to plant and walk away from, but if we actually start to rethink the different functions that we can get from these riparian buffers, we actually will generate far more ecological benefits. Even trickier still was the nitrates issue. So nitrates, as you know, is an area that is very hotly debated in the news. And I think when we think about what Brandon did in his research, he gave us a lot of reality checks about how complicated that story really is. So even with riparian management in place, we've learned that things like drains circumvent your best efforts. We can plant as much as you want, but if you have tile drains and open drains and low source areas, nutrients will enter these waterways regardless. And so what he was able to trial is one of the first in Canterbury, this tool called the bioreactor, which essentially harnesses the power of the microbes. You basically add carbon, the carbon that was lost from deforestation, into these key hotspot areas, and you allow residents time and to build so that microbial communities are converting nitrate into nitrogen gas, a permanent removal of nitrate from the system. So he tested that edge of field tool with other potential tools that we have in our toolbox for managing nitrogen. And I think what he was able to show is that nitrate is really complex because it really is complex. And that understanding the scale and the sources of nitrates is really important in this conversation. That we need to guide this rehabilitation tools in a way that addresses the fact that nitrogen can do certain things. Our nitrogen cycling can be enhanced through carbon additions, but we need to really trial that more and at scale. But monitoring, much like all of the restoration studies around the world, is really super key to answer that question. Okay, so this is something that we've been now reflecting on over time. If we think about what we were able to achieve in the CAREX framework, I'd like you to think about each of these boxes being a different site in our experiment, and the color intensity is kind of where we started. Some sites were high green, really had some restoration in place, they were doing the best practice possible, and other sites, like Project 9, had nothing. We were able to undertake things like riparian planting to enhance the condition of those waterways, do monitoring at different scales, and really use this information and really coordinating these different sites to see whether or not we could do these trials embedded within the community. So in this case, in a few sites, we did riparian planting. As we continued on, we were able to do trials such as Katie's experiments on weeds, and also monitor things like nitrates. We were able to then also undertake some sediment removal trials in a subset of these sites, all while monitoring the same attributes over time and over space. And this went on year after year, and so what we were essentially doing was learning from one year to the next, in some sites trying to apply knowledge from one site to the next and show through example and monitor at different scales to see whether or not we were making a measurable difference. And so year 5, we now have this data set of actions that were undertaken in different combinations. And I have to admit, there are no data sets that I know of in stream restoration that can offer this sort of picture. And when we think about the actual along the way, while we do have the scientific data, we have the social piece of a story of actions being undertaken across the catchment in ways that we would never have really predicted. And that we think is a reason, one of the reasons why CAREX has been a really interesting project. We have community engagement pretty much all the way through at most of these sites, and now these restoration actions are being undertaken in different places and taken up. And so as we still continue to do the science part, there was this thread of translation that was happening. And so if you go to our website into our Figshare page, the sharing of the information was our mechanism for building that trust along the way. And so this is what ECAN and the industry and farmers are referring to and are asking for. And so it's really had us thinking about what do we consider as success in terms of stream restoration, and where do you go from up here. So many of you will know this site, and so for people that haven't been to a CAREX site, I just wanted to show you this time series. This is one of our 9 sites, and this is the sort of thing that we were able to undertake. So this is a stream. You'll have to believe me that it was there. We sort of used our monitoring data and our conversations with farmers to drive the actions on this site. We pulled back that hedge and realized there was a stream there, in fact, and that the management regime resulted in a lot of that sort of sediment being piled up on one side. So this is sort of the reality of it. The community got involved in the planting, and we then implemented we picked plants based on those traits, so CAREX sector to try and outcompete the weed issue. And we've been monitoring over time. So this is where Katie has done her research. We were strategic in trying to put shading on one side to get shade boosted over the waterway in the short term, and then had one side more narrow because that's our negotiations with the district council about having a contingency for the future. And over time, we've been watching this place grow while also benefiting from that journey along the community. At a big picture, so that watershed that I showed you was actually downstream from a 1 hectare wetland, and this is now the place for a number of really interesting research, including looking at sediment traps, using the waterway and the wetland as a place of outdoor engagement, and 3 different University of Canterbury either courses or programs use this wetland to think about the water quality and ecological impacts of farming and in the Te Waihora catchment. So that's just one. So the question, we're now left with this question, or grappling with the question that we still haven't solved the problem of restoring whole ecosystems. And that's kind of the research that I'm really excited about today. We know that we've only just started the journey. It's going to take a long time for plants to grow and for these negative impacts of agriculture to really truly be mitigated and removed. And we've joined and are supported by the Biological Heritage National Science Challenge, because the question of why is it taking so long for these ecosystems to recover is something that's actually a national question. We've noticed that these food webs are dominated by really unhealthy, kind of undesired taxa, snails and worms and that sort of thing. And so how can we use and combine and test theory to try and find additional tools for biological recovery? So it's not just sediment traps, but maybe combinations of things. And so I won't go into it, but just invite you to reach out to Izzy to hear more about her research. She presented this at ABC. And then another question that this connects to is how do we, if we're thinking about the food webs, we also really think about species like kakahi. So there's a lot of things in the food webs in these agricultural waterways that we still don't have answers for, and some fundamental research needs to continue on in this space. Okay, so I've given you 2 examples, very different, an urban one and an agricultural one, around how you devise new methods for trying to do restoration and answer questions. And a lot of it has to do with identifying risk and uncertainty. It's measuring things that haven't been measured, trying to find new methods, and hopefully that gives you some ideas of where this might go. So I just wanted to take the last few minutes to talk about the bit around scaling. And what was really clear is that we talk a lot about doing ecological restoration, and a lot of the time as ecologists we spend our time thinking about those ecosystem components. Are we restoring an ecosystem as maximum as possible? If we want to achieve long-term sustainability, are we being informed by past and present? And when we look at the reviews and the reflections of the restoration community to date, a lot of the biggest gaps have been in our ability to engage and benefit society. In particular, if you look at some of these two studies in particular, some of the best cases of ecological restoration have been in examples of river and stream restoration, but they've also been in communities and in situations that have engaged First Nations or Indigenous communities. So I'll give you two examples of how we've done this component. The Headwaters stream bit. So remember I talked about me and Jake studying all these little streams, and in that work we realized when we synthesized the literature, a lot of people were measuring things, but they weren't doing things at the same time or using the same methods. We tried to leverage that power of the small stream community, and in this past year this community collected more than 200 samples all over the world and measured the same thing in the same places. This was one example of if you collect and harness the collection and measure the same things, we were able to then generate carbon cycling rates that show that intermittent streams generate more CO2 than permanent streams. And if you include those in our daily estimates of carbon cycling, this augments our estimates by up to 10%. Normally, and in a lot of the climate change cycles, intermittent streams aren't even part of the mix. And so this is one example of if you want to stay in the stream world or in the science world, harnessing the collective to understand how these small streams are contributing to larger processes can be possible. Secondly, when you look at the research around the world about examples of freshwater and stream river restoration that have achieved ecological outcomes, a lot of those examples have been because they've engaged Indigenous communities. And I think in these examples, if you look at these papers, they refer to river restoration practices in the Pacific Northwest of British Columbia and Canada. And thirdly, I didn't even talk about the UN level, but this was one pathway for me to see what science is being used to make decisions and policies. And in this community, it was really clear that scientists and decision makers were not really talking. And so it didn't matter that we had these really great syntheses or that we knew about the science. If it wasn't digestible at the this policy making avenue, it probably didn't get taken up in these syntheses. So I learned a lot about the language that's used or how decisions are made at these national levels, and how policy, like the national policy statement and and lots of these things at a national level, are informed by the kind of science that's published. If I can make one recommendation, it's publish your data, because we were really limited in synthesizing data for the Asia Pacific based on whether or not we could actually find it. And so in being part of that community, it's been great to realize that actually we need to grow more capacity at all of these different levels if we actually want to try and make something small be relevant at a national or international scale. So I just wanted to end by saying all of this has been work that's been happening here in New Zealand, thinking about if we want to do science different, or if we want to try and address questions and solve problems, we need to take a more inclusive approach. And so you'll recognize quite a few Canterbury members and biology members, where a year ago we got together to talk about, across the science disciplines in Aotearoa, how can we achieve better science outcomes, and what kinds of things need to change individually, within research teams, within institutions in the science landscape, to try and make sure that the science that's being generated actually truly is the kind of science that we want to be addressing the problems that we need solving. So I'm not going to convince you about this. This has been a rich discussion over the last year about why diversity and inclusion is important. All I'll say is just be a good scientist and ask yourself, is the science that you're generating answering the question? Is it useful? If you're getting public funding, can you talk about it with the public? Have you talked to another type of scientist and just ask whether or not they're offering a different way of doing something that might actually be more useful in the context or the problem that you're trying to solve? And maybe the outcomes that you will generate from those sorts of conversations and collaborations will be far greater than you might expect. So with that, I just wanted to kind of return back to my home and my homeland, and thinking about you know, there's a lot of big problems, but for me, it's about thinking about these headwaters, thinking about this waterway in particular was a quarry in a place that was pretty much an earmark, and nobody went to it in Toronto. It's now the hot spot for all the hipsters. It's also embedded. This is a constructed wetland, and all the tributaries have been planted. What used to be the quarry for the city is now a place where people can buy vegetables, and that's exactly where I'll be focusing my work. Really thinking about small streams, thinking about the communities that we need to work with, and just connecting what we do in these small places to larger discussions. So this is what I'll be doing, developing these relationships between microbes and the rest of the trophic level, embedding restoration trials with communities. And when it comes to just trying to think locally and globally, it's hard, nobody knows how to do it properly, but we just have to do it anyway. So with that, thank you very much. The final thing, surround yourself with rainbows. I just wanted to acknowledge FERG because this is a great place, a lot of diverse ways of thinking and doing, and if you aren't constantly surrounding yourself with different types of people and ideas, then you're probably limiting yourself. So just thanks to FERG for giving me a home, and I'm happy to take questions. Plug for CAREX. Hey, John.
Yeah, I just wondered if you've done any experiments or anything looking at the cost effectiveness of some of the restoration?
Yeah. So it's really different, right? Because for every issue, it's a different way of measuring. So one example I can think of is Katie's work. It's very linear, like plants are present over, the way that rates are kind of distributed are farmers will pay a certain amount of money to have their waterways managed. And so if you take that amount that they pay per year, then you can kind of divide that by meter or by annual rate. And so Katie basically compared the time and the labor and the cost involved with her trials to published rates for how much people were paying for over time. And what she found was that for something like, that sounds really hard, CAREX planting and managing and using weed mat over 2 or 3 years, intensively trying to get the CAREX to combine, it actually over 10 years evens out to be to the mechanical clearance rates, and the only thing that was more expensive was hand weeding because you're it's manual labor. And so so that's something. So when I talk about that with economists, this is why I like talking to economists, because I ask them what do you need to measure? What do you need to know? So a sediment trap, for example, and so Christy will know this better than I. How many hours did the digger take to rebatter? And so it's really kind of thinking about, yes, we're doing the science, but we also need to keep track of how long things took, so that when the community asks how much did it take and make the decision about whether or not it's doable, then they can make the informed decision. So a lot of the time we talk to practitioners and digger drivers, like we know the good ones now, by asking them how long will that take you, how much will it take, and then we can use that, because we've basically worked with the individual farmers to write their own grants, because we've done the monitoring, but the funding for the earthworks and planting goes to them. And so I think we have those numbers now. So if there are economists in the room that want to take over that, but that would be an example of what Jim Boyd talks about as an ecological endpoint. So what we generate, can we offer some bit of information in a form that would be usable for something like an economist? But so where that's possible. Same thing with the bioreactor, we count how long it took. So we know generally how much earthworks was involved and how much time was involved to do it. And so hopefully if someone wants to do the maths around that, that's available. And that's another reason why we co-develop with industry, right? We ask them, do you have this already? And they probably know really well what those numbers are, but what they don't know is how well they work. So that makes it a really good partnership.
I'm just interested in your Canterbury work.
Yeah.
And between the local farmer and the global, you've got the local authorities.
Yep.
How do you work with local authority levels and
Like-
decision making about that?
Like ECAN or?
Well, you can and the district council.
Yep, so we've had really good relationships with both, and just in different ways. So we've had great relationship with the Waimakariri District Council and the river engineers, in particular, Greg Bennett. He basically does all of the water management, and so we basically, it's sort of this co-development process where we're interested in these issues. What are your issues? What are your tools? And with someone like Greg, reg, he has a universal consent to dig holes in waterways. So that overcame a barrier for an ecologist because we didn't have to muck around with that. We could just do the trial, and we stood on the side of the bank together and did that. And then it just kind of, and then as well with the farmer. And so the farmer can then watch as well and figure out whether or not they can manage. The same thing with the Selwyn District Council. So it took them a little bit longer to get to the show, but you know, and organize themselves, but they've been really supportive. They've funded planting in the wetland. They're now thinking about doing a constructed wetland around Coast Ford, and so it really has been this like two-way conversation about what we've learned, and just different districts have been in different ways. And I presented some of this work to the Ashburton Council, and the mayor came right up to me and was like, I noticed Ashburton District Council is not a logo on your thank you slide, and we want to change that. So it just, people want the information, but just at different times. So we've just gone to where the energy has and tried to, use trials, fit trials in while kind of monitoring alongside. Sorry, yeah, Mary, kia ora.
E hara tēnei I te pātai, engari he kōrero ke he mi ki a koe, I runga I tō kaha whai, I te tikanga rua, I te wharawānanga nei. Just a special shout out to Catherine. We're going to really miss her. She's been one of our key operators in developing bicultural competence in science practice. And her leaving you means that some of you guys have really got to step up. So I got my eyes on you. So I just really would like to thank you, because our office feels a great loss at your party. Kia ora. I have to go.
Kia ora, Mary. I'm not sure whether I really want to say too much. I was actually decided that I was going to tell a story. I will continue to tell the story. Last year we were at a conference, I think it was last year or not the year before, and someone was introducing their colleague who had just left the CRI, and they said that this person had gone to the dark side, and they had just left the CRI to go and work for an organisation that was involved with the dairy industry, and the person who was being introduced stood up and started to talk and said, I haven't gone to the dark side, I've just gone to the cold face. And that really resonated with me, and I think that's what Catherine's been doing for us here, and really can't express how much we thank you for that.
Thanks.